You know, you can think of your body as this giant, bustling city, home to trillions of tiny inhabitants. Now, most of them are totally peaceful, just going about their day, but lurking in the shadows are some of microbiology's most wanted criminals. And today, we're going to open up their case files. So, here's a thought. What if bacteria actually had rap sheets? It's a fun way to think about it, right? It lets us go undercover into this microbial underworld. We're about to profile these pathogenic bacteria not just as organisms, but as actual suspects, with known hideouts, distinguishing features, and a whole list of crimes they've committed. All right, first up, we've got a whole crime family known as Streptococcus. I mean, this gang is everywhere, and its members are responsible for a huge range of offenses. Everything from minor infractions to life-threatening felonies. So let's meet some of the key players. Our first suspect is Streptococcus pneumoniae, but on the street, it's known as the lanced-shaped shadow. And that nickname, it comes from its signature look under the microscope. These pairs of oval cells that have these really distinct pointed ends. This guy is a major player when it comes to respiratory tract crimes. Okay, let's break down its rap sheet. First, its appearance. It's gram-positive, which just means it holds on to a specific purple stain in the lab. Its MO, or modus operandi, is something called alpha hemolysis, a partial breakdown of red blood cells. Its main distinguishing mark is this protective capsule, which we can spot with something called a quelling test. But, you know, every criminal has a weakness, and this one has two big ones. It can't survive in bile, and it's super vulnerable to a chemical called optochin. And that vulnerability, that's going to be key. And here's the kind of damage it does. Its criminal record is, well, it's extensive. It ranges from pneumonia, which is its namesake crime, to meningitis, a really serious infection around the brain and spinal cord, all the way down to those common, annoying ear and sinus infections. This is one versatile offender, for sure. You know, forensic microbiology is all about telling suspects apart. So here, we see the shadow compared to some of its less aggressive relatives, the viridans streptococci, who we can call the dental delinquents. Now, they both cause that same alpha hemolysis, but here's the crucial difference. Our primary suspect, acidemia, is sensitive to optochin and it dissolves in bile. The dental delinquents? They're not. This is exactly how the lab catches the right culprit. Next up in the strep gang is a real specialist, Streptococcus agalactiae, also known as the newborn nemesis. And as that name suggests, this bacterium poses a very severe threat to newborn infants. Its rap sheet tells us exactly why. So unlike its cousin, this one likes to arrange itself in chains, and it has a totally different MO, beta hemolysis, which means it completely destroys red blood cells. But the real key piece of intel here is its hideout. It's a normal, harmless inhabitant in about a quarter of adult women. But that's how it gets the opportunity to infect a newborn during birth, which can lead to really severe diseases like neonatal meningitis and sepsis. Okay, now we're going to leave the strep gang behind to profile a completely different kind of criminal. This one isn't so much a specialist as it is a hardened survivor, a real lone wolf that just thrives where others would perish. Let's talk about Enterococcus. We call Enterococcus the hospital hard man because it's just notoriously tough, and it's a frequent cause of those hospital-acquired infections. It normally lives in our gut, totally fine, but when it gets into the wrong place, it uses its incredible survival skills to cause some really serious trouble. I mean, this thing's toughness is legendary. It can grow in high salt conditions, it can withstand a huge range of temperatures, and it can survive being dried out on surfaces for long periods of time. This resilience is exactly what makes it such a huge problem in hospitals. And to make matters worse, it's developing resistance to some of our most powerful antibiotics. So, how does the heist actually go down? Well, it's often a really simple chain of events. The bacterium gets from its normal home in the gut onto someone's hands, then it gets transferred onto a medical device, like a catheter. That contaminated device then gives it a direct express route into a vulnerable area like the urinary tract, and boom, you've got a nasty, hard-to-treat infection. All right, our final group of suspects are the specialists. The Neisseria family has two major pathogens, and believe me, they are very, very particular about their targets and their methods of attack. Meet the Neisseria duo, nicknamed the kidney bean killers because of their unique shape, They look like these little pairs of kidney beans. Now, they look almost identical under the microscope, but their crimes are very, very different. We've got Neisseria gonorrhea and Neisseria meningitidis. So, how in the world do we tell them apart? Well, we basically interrogate them with a simple sweet tooth test. Turns out, both of them can consume glucose, but only N. meningitidis can also consume a second sugar, maltose. This simple biochemical test is the smoking gun. It lets the lab know whether they're dealing with the culprit behind gonorrhea or the one that causes meningitis. Let's take a look at the weapons that N. gonorrhea uses. Its most important gadget is its pili. You can think of them like tiny grappling hooks it uses to latch onto our cells. It also has special proteins to disable our body's alarm systems, and an enzyme, IgA1 protease, that literally shreds our antibodies. It's a highly evolved set of tools for invasion and evasion. So, we've profiled the gangs, the lone wolves, and the specialists. Now let's see how science puts all these clues together to make a definitive identification. It's time for the final police lineup. And here it is. By looking at these key identifiers, like sensitivity to a chemical, the result of a specific lab test, or the type of crime scene they leave on a blood plate, microbiologists can definitively point to the guilty party. From the optochin weakness of pneumoniae to that unique CAMP test for agalactia, each one has a tell. This is how we know who we're fighting. And all of this brings us to our final thought. We've seen the weapons, the MOs, and the hideouts of these microbial criminals. But here's the thing. They are constantly evolving, developing new tools, new ways to resist our drugs. So the crucial question this leaves us with is this. In this microscopic arms race, are our scientific and medical defenses evolving fast enough to keep us safe?